All right, so my group, we do both experimental evolution and some of this theory, and usually I talk about the experiments, but in keeping with the theme today, I'm going to talk about theoretical work done primarily by Ben Good, who is sitting right there. So he really should be giving this talk, but because I'm older, I'm giving the talk. So anyway, direct your questions there. Yeah. All right, so this is the first time I'm talking about this stuff, so feel free to interrupt if I'm more incoherent than usual. All right, so basically what I'm going to talk about is trying to understand what you expect genetic diversity to look like in the presence of pervasive natural selection that acts sort of simultaneously at many loci across the genome. And so, this comes up in a lot of contexts. One example that we've seen in our experiments is that we run long-term evolution experiments in budding yeast, and we know that these populations are experiencing lots of selection at lots of sites across the genome, and we can sequence a bunch of individuals from within these populations. And here's an example where we sequenced a couple dozen individuals from one of these evolving experimental populations, and we can characterize the genetic diversity that we see in those samples of individuals that we sequence, and we can summarize it by a genealogy. Here's one example. And ideally, and this is the sort of data that one might collect in a natural population, and ideally what we would like to be able to do is look at this kind of genealogy and see what signatures natural selection is leaving in that data and maybe infer something about how strong selection is, how broadly it's acting from this kind of data. And you see similar things. Here's a tree of some influenza sequences drawn by one of our collaborators on this project. I'm going to tell you about Richard Nair in a paper last year. And we see certain characteristics of these kinds of genealogies in the presence of pervasive selection acting across the genome. Things like the skewed branching, which you see in both of these trees, multiple merger events, which are not really characteristic typically of neutral evolution or even many kinds of demographic scenarios. But the problem is that it's really difficult to actually do anything with this kind of data at the moment, because we really have a shockingly, at least shockingly to me, limited understanding of how we expect this kind of pervasive selection to shape these genealogies. So we don't really know. I can't tell you, let's have some model of what the selection pressures are, what the mutation rates are, what the population size is, and what would we expect these kinds of trees to look like. And in the absence of that, it's really impossible, or at least very difficult, to make anything from this kind of data. All right. So what we did, I really should say what Ben did was, and what I'm going to tell you about, is a project of looking at one very specific type of pervasive selection and asking how it influences the kinds of diversity that we would expect to see. And before I get into that, for many of you this will be completely redundant, but I just want to take a minute or two to highlight what the essential problem is with the existing methods and why we need to do some work to understand these kinds of questions. So, the standard methods for looking at this kind of, data about diversity within a population, I mean, this is obviously way too big for me to summarize in a couple of minutes, but the basic idea is that what we do with something like neutral coalescent theory is we imagine some sample of individuals from within a population, and we calculate the probability of some particular ancestral relationship between those individuals, basically some possible genealogy. And of course, many of you know that there's all kinds of things we can say about these genealogies. So if we imagine sampling a bunch of individuals from the present, these two individuals might have had a common ancestor in the recent past. These two individuals had a common ancestor back at this point in the past. These two individuals had a common ancestor way back in the more distant past. And based on some assumptions about, let's say, what the population size is, what the demographic structure of the population is, we can say quite a lot about what we expect this genealogy to look like. And then what we can do is we can imagine that mutations occur as a Poisson process across the genealogy. And so we can basically compute the probability there's some genealogy, and then mutations just occur randomly, and these mutations then generate a pattern of diversity that you would observe in sequence data. So here's an example. this is the sequence of this individual here, and it has this green and this blue mutation. This individual here has the green and this yellow and the blue mutation, and so on, right? And the key thing about this is, and the thing that sort of makes this all work, is that we can separate these two processes. We compute the probability of a genealogy, and we compute the probability of an observed pattern of mutations given the genealogy. And because we can separate these two things, this whole approach works. And I'm not going to get into the details, but the two sort of key basic results that will be important for what I'm going to tell you in the rest of the talk are that if we think about the typical time since the last common ancestor of two individuals, if we imagine some population of some constant size n, okay, or alternatively some more complicated scenario but some effective size n, the probability in any given generation two randomly selected individuals share a common ancestor is just 1 over the population size. Right? The probability you just pick two people at random from a population, they have the same parent, is like 1 over the population size. So the typical time since the common ancestors will scale with the population size, and that means that the average diversity will scale with the population size. Okay? And here I'm just talking about this diversity pi, which just reflects the average number of differences at the sequence level between two randomly sampled individuals. So for example, this individual and this individual differ at one site, so that pi is 1. This individual here and this individual here, if you look at that carefully, they have 6 mutations that are different between them. So the pi there is 6. And of course, there's a whole distribution of this diversity within the sample, but on average it's going to be proportional to n. And we're also going to talk about frequency spectra, which is basically saying how many mutations are there that are present in I individuals out of our sampled, I think there are about 12 or 13 individuals here. So for example, this mutation here is present in only this individual. Okay, so that has an I of 1. This mutation here is present in 2 individuals, so that's an I of 2. This mutation is present in 7 individuals, that's an I of 7. And we can calculate in this model how common we expect mutations to be that have different frequencies. And we expect rare mutations that are present in only one individual to be more common, to be more frequent than mutations that are present in many individuals. Okay? This is all totally standard stuff. Okay, but like I said, when we have pervasive selection acting across the genome, this basic separation between the genealogy and the mutations on the genealogy breaks. Because we can imagine, let's say this is a deleterious mutation, we can't just say, okay, this happened randomly on this branch, because the fact that there's a deleterious mutation here makes it less likely that this lineage has this many descendants. Okay? So how are we going to address this? So the basic framework that we use, the idea behind all of the sort of methods that I'm going to tell you about is that we keep track of not only the ancestral relationships among individuals, but also the fitness of each ancestral lineage. So the idea here is when there's selection going on, there will be some distribution of fitnesses in the population. Some individuals will be more or less fit than others. You can imagine sampling and sequencing a few individuals and they will have random fitness. Okay? Now think about their ancestors. Right? You can think about this just like, this is like a little thought exercise for those of you in the room. How many of you guys have kids? Okay, how many of you guys don't have any kids? All right. So some of you guys are just young, but some of you guys will never have kids, right? Right? It's like, that's low fitness. You might have deleterious mutations, something like that, right? How many of your parents... full of mathematicians. Exactly. But how many of your parents have kids? Right? Like obviously all your parents have kids, right? And your grandparents, not only do they have kids, but their kids have kids, right? So if you think about it, like as we go back in time, you are lower fitness. I mean, some of you have many kids, right? Bless you. But on average, right? Like if we take all the people in this room, all of the parents of the people in this room will be more fit on average than all the people in this room, right? And if we think back further in time, that gets more and more true, right? So like our ancestors, the ancestors of the individuals currently present in the population will be a sort of fitter than average subset of that ancestral population. And the further back we go in time, the fitter and fitter those ancestors were, right? And because they're descended from a fitter and fitter subset of the population, if we think about the genealogical process, right, they're sort of you're more likely to share common ancestors. Because, many years in the past, there were only a very small fraction of the population that everyone is descended from, so they're very likely to be the same individuals. Right? And we can sort of quantify this, like if we go back in the past, we trace these ancestral lineages, they're a bit fitter, they get even more fit, and if you look way back in the past, everyone's descended from these sort of most fit individuals, right? And so we try to understand this. Okay. Now, obviously, I can sort of say this intuitively and talk about your parents, but let's try to make this a little bit more concrete. Okay, so all of this, this business that I'm telling you about here, I mean, this applies regardless of the type of selection that's going on. But if we want to actually make quantitative predictions about this sort of stuff, we need a concrete model. And so for this purpose, we're going to focus on one specific type of selection. So we're just going to focus on pervasive purifying selection that acts across the genome. Okay, so we're basically going to say, all right, there's a lot of mutations that can happen that are bad. All right, and obviously there's lots of other stuff too, but this is like, we know that this is always the case, right? There's always lots of mutations that are bad. And so we're just going to focus on what effects those mutations have, okay, and ask how that would affect diversity. And we can think about beneficial mutations and so on maybe some other time. So the really simple, dumb model that we're going to look at, we're just going to imagine a population of constant size n with some mutation rate u, okay, genome-wide mutation rate u. And we're just going to say mutations, each mutation that occurs, we're going to imagine a sort of infinite sites model. So each mutation occurs at some unique site in the genome, and it has some fitness effect drawn from some distribution, and there's some recombination rate. Okay. And this model is completely unsolved. We have no idea what genetic diversity is going to look like, and I'm going to actually tell you an even simpler model because we're not even going to solve this one. We're going to solve an even dumber model. All right. This is the even simpler model. Okay, we're going to imagine that that mutations have one of two fitness effects, either zero, okay, they're neutral, or they have some fitness cost s. So there's some deleterious mutations. And so then we can just divide this mutation rate into the neutral mutation rate and the deleterious mutation rate. That's it. Okay. This is like about as simple a model involving natural selection that you could possibly write down. Okay, later we'll talk about the effects of recombination and distributions of effects right at the end of my talk. But for now, we're just going to think about this very, very, very simple model. And we're going to imagine a recombination rate of 0. Okay, so we're going to be looking at within a short distance scale within a genome where recombination is very unlikely in a sense that we'll get to right at the end. Okay, so all right, that's the model. Is this clear what I'm talking about? All right. So within this model, we can do this kind of tracing of the genealogies of our ancestors through the fitness distribution within the population in a very simple way, right? So we have these deleterious mutations. They have fitness effect S. So if we think about the distribution of fitness within the population, some individuals will be very fit. They don't have any deleterious mutations. Some will have 1, 2, 3, and so on deleterious mutations. This is known as a mutation selection balance. It's set by mutations driving you this way and selection sort of pushing you that way, and so you get some steady state distribution. You can imagine, let's say you sample 2 individuals from the population and you think about their ancestries. We can compute the probability, say, for example, at some time in the past, this individual descended from a more fit ancestor, which had this higher fitness. And then maybe sometime even more in the past, the ancestor of this individual was descended from an individual that had higher fitness, and maybe then this. Now these 2 individuals at this point in the past had the same fitness. Maybe they had a common ancestor here that had 1 deleterious mutation, or maybe not. Maybe this individual descended from an individual that had 0, and so on, and eventually they have some common ancestor. We compute the statistics of all this stuff and figure out what the genealogy looks like. Okay, and this is known as the structured coalescent. It was introduced by Hudson and Kaplan 20 years ago now. And a lot of work by them, and since by some of us, has worked out what we expect the diversity to look like, how we expect this kind of selection to influence patterns of sequence diversity.
You're big enough population size that you don't lose any.
Yeah. Well, so I'm still in like, yes. But what we're going to talk about is when we're not. The whole point of this talk is going to be what happens when that breaks down. Yeah, but for the moment, yes. For the moment, we're going to assume a large population and where this distribution is fixed, all right? Until the next slide. All right. So
Michael, can you go back to the previous slide? So if you remove the recombination in a simple model, why isn't that sort of essentially just sort of structured coalescent with infinitely many?
That's what it is, yeah. It's a structured coalescent with, yes, that's all this is.
The first model is that, right? It's a structured coalescent with infinitely many.
Right, which
actually depends on distribution.
Yeah.
Like for distributions that don't have a harmonic mean, there is no structured coalescent.
Yeah.
But it's in terms of the, you do have this sort of migration type thing.
Right. I guess what I shouldn't say is that when I say it's unsolved, what I mean is that you can sort of write down a structured coalescent as long as this has a harmonic mean. If it doesn't have a harmonic mean, that doesn't even make sense. However, you can't actually compute anything. Okay, and as we'll get to in a minute, the structured coalescent breaks down in many other ways as well. Okay, but we're coming to that in just a sec. All right. So the thing that's sort of well known and that like you'll see in textbooks and that everyone sort of talks about is known as background selection. Okay, and this is a sort of limit of all of what I've been talking to you about so far when selection pressures are really strong. So if you think that the selection pressures are really strong, okay, then all of y'all, you may have some deleterious mutations, but you must be very recently descended from individuals that don't have any deleterious mutations, because if those deleterious mutations are so bad, like your parents couldn't have even had kids if they had any. Right? So your ancestors are very recently descended from this class of individuals that has no deleterious mutations. So we can think, if we sample individuals very quickly, they're descended from these individuals that don't have any deleterious mutations. And then the genetic diversity within the population is characteristic of, well, these deleterious mutations happened during this sort of most recent phase of the ancestral history. Right? If you think about the three individuals, they very recently descended from this class of individuals that had no deleterious mutations, and then you have an essentially neutral process within this class. Okay? This is called the delay time, this is the coalescent time, and when selection is sufficiently strong, this time is short compared to this time, and basically what you have, all right, since there's these great boards, I'll just write up, you have okay, that the
one is a subscript and the other one's.
Yeah.
Yes, like this. This is the actual, yeah, thank you. I'll write that. Right? Very good. So, yes, one is 2.7 and the other is a subscript. Right, so you can work out in mutation selection balance, this class will have a fraction of the population of e to the minus ud over s. So this is the population size of this class. So the neutral coalescent process within this class will be it'll be just a standard neutral coalescent with this reduced effective population size. And the diversity that you see within the population will be characteristic of this, plus these little branches right at the end. Okay? And this is formally exact in the limit as ns goes to infinity. Okay? But you have to sort of be careful what you mean by this. If we just take ns going to infinity, okay, while keeping some fixed mutation rate, then you'll just get a neutral population. It'll be as if selection wasn't there. If mutations are really lethal, mutations don't matter. So it's formally the limit as ns goes to 0 while taking nu to infinity in a way in which nu over ns is constant. Okay? And that gives you this result. Okay, but now as there have been various questions to describe, right, this depends exponentially on ud over s. So it is not very hard at all to imagine scenarios where if ud becomes bigger than s, okay, very quickly this will break down. And this e to the minus ud over s will become tiny, and this ne may even become much, much less than 1. Okay? And this whole distribution, okay, that we talked about here doesn't even make sense because you can't have less than one individual, right? And so this whole distribution, rather than being some fixed thing, will be some crazy fluctuating thing where genetic drift will be very important, okay? And I'm sort of just drawing a schematic here. You might imagine lots and lots of different classes with very weakly selected mutations in each class, or lots of a high mutation rate, where UD over S is large. Okay? Now the key thing is, in this sort of regime, there still exists a distribution of fitness within the population. I mean, of course, there's variation in fitness. I mean, if you crank up the deleterious mutation rate, you don't destroy variation in fitness. I mean, you still have variation in fitness. It's just fluctuating all over the place, and we can't sort of draw this situation where this sort of fixed distribution, these fixed set of fitness classes. But there's still some distribution, and we'll talk about there's some variance of this distribution, which depends in some complicated way on these underlying parameters. Okay, it's much less than the variance would be if we had stronger selection. Okay, but okay, we have this kind of scenario. We still want to predict how genetic diversity is going to look. Okay, so if you just run a whole bunch of simulations, so these are just a whole bunch of simulations that Ben ran. And what he did was he took a whole bunch of different selection strengths and a whole bunch of different mutation rates. And he showed that when this NE over here, when NE to the minus UD over S times S becomes of order 1, okay, so that's this line here where that happens in this selection versus mutation phase diagram here. In this whole regime where selection is strong or mutations are relatively weak, all of the predictions of this background selection, these predictions here, work reasonably well. Okay, this blue just means that the predictions are pretty good in a way that I'll define. Well, we'll see more precisely in a minute. And in this whole regime over here, all of that stuff doesn't work. Okay, so this is what we want to explain. That's going to be the point of what I'm about to tell you.
This is the pi of the deleterious and nothing neutral.
Right. Yeah. Sorry. And what this is, these pi. I'm going to talk about pi over pi naught. This is the average diversity that you see divided by pi naught, which is what it would be in the absence of any deleterious mutations. So in a neutral model. Okay. So if pi over pi naught is 1, then the population is basically neutral, and as pi over pi naught gets less, selection is reducing diversity. Okay. Now, we're just going to present some phenomenological results in the next couple minutes. So what we noticed with these experiments is if you take all of these data points from here, okay, and I just plot them as a function of deleterious load. So each of these points has some selection strength and some mutation rate. If I just take U over S, okay, which is what goes in here, okay, and for all the different selection strengths, I just plot them each point according to the deleterious mutation rate. I'm drawing all of the points in circles if they come from this part of the graph, and all the points in triangles if they come from this part of the graph. And what you see is all the points that have triangles, they fall on this line, where the diversity is predicted by U over S. I should have written UD here. This is UD over S. Okay, so UD over S tells me what the diversity is for populations that follow here, independent of what S and UD is. Okay, in other words, different values of S and UD give you the same diversity as long as UD over S is the same thing as predicted by this over here. But if you look at these points over here, that doesn't hold. Okay? However, there is something that does hold, which is that if you plot the standard deviation in fitness, okay, each value of UD and S gives you some standard deviation in fitness, and if you predict the amount of diversity as a function of that, all the points that have different UD and S, but that have the same standard deviation in fitness, give you exactly the same pattern of diversity. Okay? And the points that were over here in this background selection regime, that doesn't hold. Right? So you have these two different things that predict what diversity you're going to see in these two different parts of the parameter space. Okay? And this is just looking at the pi, the average diversity. If you look at the whole frequency spectrum, you see a same sort of pattern. I'm not going to really get into this in great detail, but here's an example. This is the neutral expectation. Here's an example of what the frequency spectrum looks like in one of these sort of populations in this interference selection regime, and you can compute the average minor allele frequency, or Tajima's D, or the sort of something about this non-monotonicity in the frequency spectrum. And again, what you see is that all the populations in this interference selection regime, if you can predict, let me put it this way, if you can predict the standard deviation in fitness, okay, you can predict the average heterozygosity, this pi, And this pi then in turn tells you what the rest of the frequency spectrum looks like. Okay? And this holds, just one sec, this holds both in these asexual populations and also with recombination in a way I'll get to in a minute. Yeah?
Sorry, is the neutral diversity done using that effective population size? Or?
No, this is just the, I mean, it doesn't matter, right? So the frequency spectrum is independent.
But on the previous slide, pi naught, for instance.
No, so this pi naught is in the absence of deleterious mutation. So you can see pi over pi naught of 1, which is over here at the left, obtains if you have either almost no deleterious load, like if you take UD to 0. Also, if you take S to 0, so this goes to infinity, you notice you get to the same thing. And in the middle, you have this reduction. And it's not, this background selection, it predicts that reduction, okay, provided that we're in this regime where this thing is large enough. Okay? So it's not that you can't get a deviation in terms of the reduction in heterozygosity, okay? But from the neutral expectation, okay? Even here. So this is, yeah. Okay. All right. So what does this predict? I have like 10 minutes, is that right?
Yeah.
Ballpark? Okay. All right. So it turns out that if you think about these models, and Ben, did long supplementary material, which y'all can look at if you're interested, of this paper that we just wrote on this. Just as there's this background selection limit, which becomes exact as you take ns to infinity while holding this thing constant, okay? There's also another limit of this model where you can predict diversity exactly, or at least, yeah, a little more complicated than that. But there's another sort of simple limit of this model that emerges in the sort of opposite limit, where ns goes to 0, okay? And obviously, if you just take ns goes to 0 without doing something else, you get a neutral population, same as in the background selection regime. But if you take ns going to 0 while holding the standard deviation constant, okay, so you take ns going to 0, but nu going to infinity to keep the standard deviation and fitness constant, you get some other limit of the model known as the interference selection limit, or the infinitesimal limit, as we're calling it. And this is sort of analogous to quantitative genetics, right? This is exactly the usual quantitative genetics limit, where we imagine that the phenotype, in this case fitness, is caused by a sort of effectively infinite number of a sort of very weak effect things. It's a sort of standard assumption in quantitative genetics. You have lots and lots of loci affecting the trait. Each locus has an infinitesimal effect on the trait. Okay, here just the trait is fitness. That's what this limit is doing. And we're saying we're holding the variance in fitness constant. Yeah?
So your parameters here that you have to play with are UN, UD, and S, right?
Yes.
So how does sigma arise out of those? Because
so that's a whole complicated story, which I'm not going to get into. But Oskar Halacek made this very nice model. Okay, so first of all, there are really only two parameters, okay? Because UN doesn't affect any of this, it just scales the overall everything. So really, we just have UD, so that's why I'm been sloppy here. These U's are all UD, and then this is S, and that's it, right? So we have NUD and NS, that's it. And using this model introduced by Oskar Halacek in a couple years ago, I guess 2011 now, you can calculate how this variance in fitness depends on UD and S. Basically, it goes, well, it's complicated. Ben, I don't know if you want to say anything more specific about it, but it's some complicated function which you can calculate.
Okay.
Okay.
Only weekly depends on the population.
Yeah, and it's weekly. Yeah, it's. All right, I don't want to say anything more about it because I don't have that much time. All right, so the upshot is. So in this whole, and there's some boundary in between, okay? And this boundary is, loosely speaking, this same boundary that I've been talking about before when NE times S is 0. This is where the distribution of fitnesses starts fluctuating a lot, okay? Exactly what this boundary is a little tricky, but roughly speaking, that's what it is, okay? So the idea that we had was that, okay, in this background selection limit, we had some formal thing that was true way out here in this limit. But it seems to work pretty well throughout this distribution, and with the structured coalescent, we can work it out. So what one could imagine is you take some population that has some variance in fitness over here, okay? it reduces to some interference selection limit with the same variation, with the same variance in fitness, if we sort of go off in this direction. Okay? And that might be a good approximation for this diversity. Okay? And sort of more generally, because we see, okay, in all of these plots, the standard deviation in fitness predicts everything sort of throughout this regime, we hypothesize that any two populations that have the same variance in fitness in this regime, okay, have the same patterns of diversity. That's what these simulations are telling us. Okay? So what we could do, we could imagine taking a population that sort of lives over here, compute another population off in this limit that has the same variance in fitness, okay, and predict the diversity that way. Okay? The problem is you can't actually solve this limit and predict the diversity like you can for this background selection limit. But what you can do instead, okay, is you can say, all right, this population has this variance in fitness. We know that populations that have the same variance in fitness in this regime have the same patterns of diversity. So let's approximate the diversity we expect to see in this population here with a population way over here on the boundary that has the same variance in fitness. And how are we going to do that? We're going to approximate this population as being in the background selection regime. Okay? Well, it's like a really kind of a funny thing. We're going to take a population where all of this background selection stuff is breaking down. We're going to say it's equivalent to a population with the same variance in fitness right on the boundary of this regime. And then we're going to use this strong selection to predict the diversity. All right? And intuitively, what's going on here is this is like coarse graining the distribution. Okay? The sort of inspiration for this came about thinking about the neutral limit. Right? Like if we think about these kinds of distributions, right, of the number of deleterious mutations that each individual has, right, this makes sense in the limit as s goes to 0. Right? I mean, I can talk about the number of deleterious mutations that you have even as s goes to 0. Right? But there, like, the whole distribution is fluctuating all to hell. But we know that everything is described by this very simple neutral coalescent. And the reason it works is because we're sort of forgetting about all those fitness classes, because then they're not fitness classes anymore, right? All the mutations have zero fitness effect, so we're just treating them as one big group, right? So the idea was, okay, what about for small but non-zero fitness effects? Maybe we can do the same kind of thing. Imagine you have some distribution. I'm just zooming in on some piece over here, let's say. We have all these different fitness classes. They're fluctuating all over the place, but let's just group together some of these classes as some sort of coarse-grained group of mutations, right? And so here I'm just drawing some rough schematic of that, right? 4 different of these classes I'm sort of grouping together as one class. And then I'm sort of redefining. This is my new selective effect, okay? And now many of these mutations that were previously classed as deleterious now become effectively neutral, because they just move you within one of these groups. And only some fraction of the mutations are deleterious, the ones that move you between these groups. So like in this sort of context, what this is doing, it's taking a larger, a smaller s and a larger ud and mapping it to a different model with a larger a smaller ud and a larger s. In other words, we are going from here down in this direction. Okay? Now obviously you have to do this right. Okay? You cannot group these things too broadly, because then you will be ignoring fitness effects that really do matter. And you cannot do it too narrowly, because then the distribution will still be fluctuating and the methods that we'll use to predict the diversity using this background selection ideas won't work. Okay? And it's not obvious that there is a mapping that works, okay, from this. Okay, but as it turns out, and inspired by these simulations, there is a mapping that works, and it's the mapping that redefines this thing in such a way that you preserve the same variance in fitness. Okay?
It's a way in which then both approximations
become
good?
Exactly. We're just patching it, exactly. There's this regime in the middle.
Both are sort of intermediately bad, or both are actually good?
Both are actually pretty good. So,
the talk and
yeah. Sorry. Okay, so let me just highlight the results, and this will get at like how good it is. All right, I'll just take another minute or two. So, first of all, this causes a big problem of identifiability, right? It posits that two populations that have the same variance in fitness here, but very different underlying parameters, have the same patterns of diversity, and they sort of look like this. So it actually, like, for those of you that sort of read a lot of this literature, you'll often see like estimates of diversity within populations corresponding to any S of order 1. And what this is saying is that, you know, those are populations that they could have parameters where any S is of order 1, but they're also consistent with all the other sets of parameters that have the same, that would give you the same variance in fitness, but might have very different underlying mutation rates and selectin pressures. Okay, and this is just going back, these red lines now, these are the same plots I showed you a minute ago. This is the prediction of that method that I just talked to you about. So it's really damn good. All right? Here's the whole frequency spectra. There's some problems in the middle here, okay, but these are various populations with different underlying parameters, but that have the same variance in fitness. You can see their frequency spectra are the same, and this red line is our prediction. Pretty good, except for a little problem here, which we still haven't worked out. Here are various summary statistics of that, like the minor allele frequency, Tajima's D, this non-monotonicity. And this is for recombining as well as non-recombining populations. Pretty good. All right, and the last thing I just want to tell you about very quickly is how we include recombination into this, right? So far we've been thinking about asexual populations, but if you imagine a recombining population, what we can do is just imagine, okay, you have some long genome, and imagine two sites very distant, okay? If the recombination rate if the total number of expected recombination events in the time since the most recent common ancestor of some sample is large between two very distant sites in the genome, these will be effectively evolving independently. On the other hand, if you look at a short distance scale, okay, these two sites that are very close together might essentially never recombine in a coalescence time. So they're effectively purely asexual. Okay? And so there's some balance between these two, and there's some sort of distance scale within the genome, which we're calling a linkage block. Right? And sites that are close enough together, they're sort of effectively asexual, and all that I've been telling you about is applying. And then sites that are more distant, you just have independence. Okay? And so the scale of this linkage block is the distance scale in the genome in which you will have of order 1 recombination event in the time since the most recent common ancestor. And of course, this time to the most recent common ancestor depends on the size of this linkage block, because the larger the linkage block is, the more deleterious mutations there are, the higher the UD, and that of course affects this. So you have to self-consistently figure out what this is. But we can do that, right? And so what we can predict, for instance, is how much diversity we expect as a function of the recombination rate and the mutation rate. And you can see, for example, this is simulations, and this red line is our prediction. These other lines are various predictions of some earlier work, which, as you can see, don't work as well. This is the average diversity. This is a measure of the site frequency spectrum. Okay, and we can also look at distributions of fitness effects. I don't want to get into that. But here, this is just showing you this diagram again. And what I hid is all of these different lines, these different frequency spectra, come from populations. Here's a sexual population, populations with different distributions of fitness effects, a finite sites model, a model of the type, an asexual model of the type that I was telling you about. They're all models with very different underlying parameters, but they have the same variance in fitness. They give you the same site frequency spectra, and we can predict it pretty well with all of this coarse graining idea. All right, so that's it. This is the group. This is all work of Ben Good with our collaborators Alex and Richard. Thanks.
Questions, anybody? All right.
Have you looked at other patterns of diversity besides the site frequency spectrum, like linkage related, to see if it's identifiable from those?
No, we haven't. This is an interesting thing to do. I don't know, Ben, if you want to.
Yeah, we haven't looked at it yet, but that is something interesting.
Yeah.
Can you repeat the question? What does she want to look at?
She wants to look at, essentially, all of what I've told you about is looking at each site sort of by itself. And if we look at sort of the correlations between diversity at sites as, say, a function of the distance along the genome, maybe there's something more we could say from that. And we have not done that, but it is interesting to do.
I mean, Richard and Oscar both looked at similar things, like with linkage
Right.
equilibrium, and you see similar patterns, but it's
I guess, I mean, all of this assumes that the deleterious mutations are non-epistatic, right? So for Malus-Ratchet, it's known for, I mean, this is basically Malus-Ratchet model, and here it's known that the Malus-Ratchet can be stopped, for example, by various kinds of epistasis.
It's not really assuming no epistasis. I mean, there can be a lot of epistasis. What it's assuming is that even if there's a lot of epistasis, essentially the distribution of fitness effects is not substantially different for any two genotypes. So you can have individual mutations have epistatic interactions with each other, but as long as the distribution of fitness effects isn't changing wildly between sort of nearby genotypes, then this general thing will hold. But yes, it does make that assumption, and when that breaks down, this will not hold. I agree.
So back in the 90s, Santiago and Caballero
Yes.
worked through the infinitesimal selection model. And how does your results fit into that?
I'm going to let Ben answer that because
so it's more marvelous strong recombination.
Right.
Yes, exactly. Right, and there you find it's like you can approximate it with a time-dependent effective population size, but that won't lead to things like the non-monotonicity in the site frequency spectrum and other something. So.
Can I just ask you to flip those last two slides back and forth? And
this
the
one?
one about recombinate, the one before. Yeah, so I was trying to understand how this slide here on the bottom left is integrated then into the summary slide that you show us, where you say they're all the same. There's some integration over the genome or some way to bring those together?
So what one can do essentially is you imagine you have some recombination, right? What you do is you just hypothesize that the length of a linkage block is some number, okay? And you compute the time to the most recent common ancestor using our asexual models, okay, that I described to you before. All of that, you compute what the time to the most recent common ancestor would be, okay? Given that LB, okay, that LB affects this because LB is sort of setting how many deleterious mutations are within a block, in other words, what the UD is. Okay, so you take the LB, that assumes a UD, you predict this. Okay, then you ask, is this greater or less than 1? If it's greater than 1, okay, then you reduce the LB. If it's less than 1, you increase the LB. You do that, and you find the LB where this is 1. Okay, it's not really exactly 1. There's more complicated, but roughly speaking, that's what you do. So then you have an LB, okay, and then you can predict what the diversity is using our asexual model with that LB. Okay? And that's how you make the prediction. Okay? And then you see that the diversity matches that from the full recombining simulation.
But the last slide implies a genome-wide property, and I was just wondering whether the genome is actually modeled in doing this sort of, in looking at the differences among those.
Right, so these are simulations of like a whole genome with the recombination and looking at the site frequency spectrum that just naturally arises in that full simulation.
But
the prediction comes about.
The prediction is what?
The prediction comes about, it is the prediction.
The prediction produces the genome, I'm just trying to see.
The prediction is the prediction of the corresponding asexual model with the LB defined in the way that I just described.
Okay. Is it a single value or is it distribution?
In this case, it's a single value.
I guess that's what I was getting at. There's a distribution, but you're using a single value.
But this is just an approximation.
Okay.
It's perfect.
Oh, sorry, this is for the mean. So if you average over the whole.
Yeah. Oh, yeah, that's right. I mean, obviously, yes.
I just,
Yeah.
Good. Good.
So you've talked about a regime where you have many mutations with small effect or few mutations with large effect.
Yeah.
What about a regime with many mutations with large effect? Would variance in fitness still be important?
So many mutations with large effect. So you want to go here, right? So it depends on how you take many mutations with large effect, right? If you just take S going to infinity and U going to infinity at the same rate, you will go off in this direction, right? And so that's just one of these models. Okay, it's either one of these effects, depending on how far exactly where you are.
All right. Well, it's been very interesting talking. You can talk to him more at the break, but I think we
Okay.
Probably-